The fetal pig is one of the most common mammalian dissections. Due to its state as a fetus, its prenatal functions can be compared to that of adult mammals, and a comparison of anatomies can be critically observed. Please select a lesson, and then press click here to begin.
Respiratory system.
In this lesson you will do the following. Find and examine parts of the respiratory system. Remove the lungs and examine them closer. Examine the alveoli under the microscope.
The respiratory system of the fetal pig relies solely on the respiratory and circulatory processes of the mother pig. However, the fetal respiratory organs can be developed enough to illustrate the complete respiratory system of adult mammals. Mammals are negative pressure breathers. This expression is derived from a discovery made by scientist Robert Boyle in the 1600s, who related pressure to volume. Boyle discovered that compressing a container and decreasing the volume of the gas inside would increase the pressure to the point that the gas would leak out. Press the container. The pressure inside was then greater than, or positive, to the outside pressure. When he expanded the same container's volume, he found the opposite was true, and that the gas inside would lose pressure and try to draw in air from outside. Press the container again. Here the pressure was less than, or negative to the outside pressure, causing a vacuum. Similarly, negative pressure breathers draw air into their lungs by expanding the interior volume of their chest cavities. Respiration is powered by the muscular diaphragm found beneath the lungs, and is accompanied by the external intercostals, which are rib muscles that stretch the cavity apart. Imagine that the cavity is closed for this portion of our simulation. Find and press on the diaphragm. As you can see, when the diaphragm contracts and ribs expand, the lungs draw in air from outside the body, causing inspiration. This action of inspiration is generally autonomic in function, yet breathing can be cognitively controlled when necessary, such as when a mammal holds its breath. Expiration occurs when a mammal releases its breath. Here the elastic tissues of the cavity, along with contractions of the internal intercostals, squeeze the ribs and force used air out of the body again. Press the diaphragm one more time to continue. The adult respiratory cycle starts at the nostrils, or nares, found within the snout. Here air moves into the nasal cavity of the skull. Press the scissors button, and then cut back the jaw tissue to allow the mouth to open wider. Open the mouth. The immediate roof of the pig's mouth is called the hard palate. Find and press on it. Similar to the roof of your own mouth, the hard palate is a tissue-covered portion of the skull that forms the bottom of the nasal cavity. The rough ridges are called rugae. The hard palate allows mammals to breathe and chew food at the same time, by keeping the nasal cavity open. Now press on the soft palate just below the hard palate. The soft palate prevents food from entering the nasal cavity when swallowing, because the tongue presses its opening shut. When breathing, the soft palate remains open. Press the rotary tool button. Cut the hard and soft palate away by holding the rotary tool button down. Lift away the hard and soft palate. Then press the Inside the nasal cavity is the nasal septum, which separates the left and right halves of the airway. Find and press on it. Near the nostrils, hairs grow, which filter large particles of dust. But across the nasal cavity's length is a mucous membrane that warms air and traps smaller particles. This membrane covers folded protrusions called conchae, which create extra surface area for the mucous membranes. Press on one of the conchae. Ciliated action moves excess mucus back into the pharynx, and then down the esophagus for digestion. A pig's sense of smell, using olfactory sensors, relies heavily upon the nasal cavity to operate. Locate and press on the olfactory epithelium at the extreme roof of the cavity. This tissue, lined with chemoreceptors, connects to the olfactory bulb of the brain, and is responsible for the pig's primary sense of smell. Now find and press on the circular-shaped incisive papilla, found just above the cut opening of the nasal cavity. On the right and left sides of the incisive papilla are tiny pores that lead to a secondary olfactory sensor called the vomeronasal organ. Locate a portion of the vomeronasal organ just inside the nasal cavity. The vomeronasal organ is found in many mammals, and is thought to be sensitive to scents used in triggering mating behavior, called pheromones. Let's move down to the pharynx. The pharynx is an area where the digestive and respiratory systems intersect. Air from the nasal cavity enters the pharynx, and then moves past a protruding flap of tissue called the epiglottis. Press on it. Good job. As the pig swallows, the epiglottis prevents choking by folding over and protecting the airway opening called the glottis. Lift the jaw shut. The glottis opens into the larynx, which is a cartilaginous framework housing the pig's vocal cords. Press on the larynx. Now press the scissors button. Cut the larynx away from the lower jaw, and then lift the larynx up. As the pig exhales and the larynx muscles tighten, the elastic tissues within the larynx vibrate and resonate sounds. Press the diaphragm to continue. Please make note that this vocal demonstration is merely a simulation of what occurs in a normal living pig, and that it cannot be done in any dissection. Now place the lungs to the right, and press the magnifier button to zoom in. Next press the pin button. Pin the larynx and trachea, and then press the scalpel button. Slice the larynx and trachea by pulling the scalpel downwards. Lift the ventral half. Locate the true vocal cords and press on one of them. Good job. The airway that leads from here directly to the lungs is called the trachea. Press on one of the cut rings of cartilage that help the trachea maintain its shape. Excellent. At its caudal end, the trachea branches off into right and left bronchus tubes. Each bronchus tube then divides into smaller bronchi, and then into tinier branches called bronchioles, which terminate at air sacs called alveoli. The pulmonary arteries also enter the lungs and follow each airway to its terminating capillary-covered alveolus. Oxygen within each alveolus is then absorbed through a thin membrane, and is exchanged with carbon dioxide inside the capillaries. After this transfer, the oxygenated blood then travels through veins that run alongside the bronchioles and bronchi, until they merge into one of the pulmonary veins. Press the scalpel button. Take a sample of lung tissue, smear it on the slide, and then press the microscope button. As you focus the microscope, you will see the tiny compressed alveoli. Upon expiration in an adult pig, the used air within them would leave back up through the bronchioles, bronchi, trachea, and back out of the pig. You have completed this lesson. Press the arrow to continue.